Good morning, ladies and gentlemen. My name is Zbigniew Krajewski, and in this series of films, which I have titled Phylogenetic Aspects of the Concept of Intelligent Life Design, an analysis based on information provided by the Bible, I will try to present the topic of phylogeny, particularly the phylogeny of mammals. This will be a film intended for viewers who are not necessarily involved in scientific work or in the analysis of genetic information. Due to the limited running time of the film, I will focus only on presenting the basic conclusions, using language that I hope will be understandable to everyone. More detailed analysis and explanations of the methods, tools, and relevant calculations will be presented in the separate series of films devoted to bioinformatics. Since this presentation concerns the topic of life, let us begin by defining what life is. According to the dictionary by Polish scientific publishers, life is a state of an organism consisting of a continuous sequence of processes that enable responses to stimuli and usually movement, as well as nourishment, growth, and reproduction. Of course, we are speaking here about definition of life in a very general sense, without distinguishing between conscious and unconscious life. The topic of consciousness itself was discussed in films number 4, 2 and 3 of the series entitled Questions About Everything. These are the films titled Ego and The Origin of Facts in the Scenario of Introduced Defect, as well as a model of the conscious mind. What is the most important in the defining life, however, concerns its informational nature. Imagine that it is possible to encode information about the structure of molecules in such a way that they performed precisely defined tasks at the molecular level, cooperating with one another and on a macroscopic scale, forming highly complex systems. Such an idea has already been portrayed in several screen adaptations involving programmable nanobots, but they are still nowhere near the level of molecules, let alone atoms. This is exactly how life operates, at the level of molecules and even atoms, deliberately and precisely constructing complex systems on both the microscopic and macroscopic scales, with the structure of the molecules that perform these tasks being strictly programmed. Moreover, life is capable of reproducing itself, transferring the entire code of life together with the basic machinery required to read and execute it. That machinery is, of course, the cell, which contains the program of life encoded in the long molecule, usually DNA, located in its nucleus. DNA programs the structure and thus the shape of molecules known as proteins. The programmed shape of these molecules, together with the specific regions called active sites, determines the function for which they were created. It is precisely this aspect of the program contained in the DNA molecule that we should focus on when considering similarities in the information that programs the life functions of individual organisms. Let us begin somewhat from the end. That is, with the molecules that carry out programmed functions, namely proteins. A protein is composed of smaller building blocks, molecules called amino acids. A protein is a long molecule made up of amino acids linked together in sequence. In general, 20 types of amino acids can occur in proteins. By themselves, they would form only a long chain were it not for their ability to fold into a specific shape determined precisely by the sequence in which the amino acids are linked within the protein. In the amino acids that make up proteins, we can distinguish a central carbon atom, hydrogen atom above it, and two molecular groups, the so-called amino group on the left, containing a nitrogen atom and two hydrogen atoms, and the carboxyl group on the right, containing a carbon atom with attached oxygen atoms and a hydrogen atom. However, what distinguishes each of the 20 amino acids that make up a protein is a fragment known as the side chain, denoted by the symbol R. This means that each of the 20 amino acids is identical in its basic structure. It contains a central carbon atom and amino group and a carboxyl group, but differs in the fragment of the molecule known as a side chain. The side chain may consist of, for example, just a hydrogen atom, as in the case of the amino acid glycine, or it may include an aromatic ring, as in the case of tryptophan. The amino acid group of one amino acid binds to the carboxyl group of the next amino acid, forming what is known a peptide bond, with the possibility of rotation around the bonds between these groups and the central carbon atom of the amino acid. Owing to this rotational freedom, proteins fold into complex three-dimensional structures that determine their functions and thus the tasks they perform. What will be the most interesting for us in the context of the subject of this film, however, is the manner in which proteins are encoded. Proteins are encoded in a molecule called DNA, which forms a long double-stranded molecule containing 4 bases arranged sequentially, designated as A for adenine, G for guanine, C for cytosine, and T for thymine. This long double strand is held together by bases paired complementarily, that is, in a matching manner, such that the successive bases A, G, C and T bind to bases on the opposite strand as T, C, G and A respectively. As we can already anticipate, an appropriate sequence of these 4 bases can program a corresponding sequence of amino acids in proteins, and thus program the tasks they are meant to perform. The code for successive amino acids is written in sets of 3 bases known as codons, with 4 possible bases at each position. This gives us the possibility of encoding 4 times 4 times 4 codons, that is 64 amino acids. This number is excessive for encoding the 20 existing amino acids. Therefore, several codons can encode the same amino acid. Amino acids are encoded within so-called reading frames, from a codon that signals the start to a codon that signals the stop. Thus, among these 64 possible codons, at least one must be reserved as a stop signal. The start codon is usually also a codon that later specifies one of the amino acids within the frame, and it is the most commonly methionine. We will discuss this topic in more detail later in subsequent slides. For now, we will limit ourselves to the information necessary to discuss the stage of protein encoding. The carrier of genetic code, in addition to the relatively stable DNA molecule, can also be an RNA molecule. This is a molecule very similar to DNA, but in this case consisting of a single strand with bases arranged sequentially as in DNA. The difference is that instead of thymine, another base is present here, namely uracil, denoted by the letter U. In the process of protein formation, information about the protein is transcribed from the DNA molecule located in the cell nucleus into a mobile RNA molecule known as messenger RNA, designated mRNA, which after transcription moves to the protein-producing machines called ribosomes. Individual amino acids are delivered to the ribosomes by so-called tRNA molecules, that is, transferred RNA. The protein is synthesized according to the encoded mRNA sequence, which, as mentioned, is a reflection of the protein coding sequence transcribed from the DNA molecule located in the cell nucleus. Thus, in our example, we have a sequence of codons TTA, AGT, and ACG. These codons encode the amino acids leucine, serine, and threonine, respectively. Consequently, these amino acids will appear in the chain of the newly formed protein. In addition to nuclear DNA, which encodes virtually all protein functions in mammalian organisms, there also exists DNA that functions independently of nuclear DNA. This DNA encodes 13 proteins in structures called mitochondria, which are responsible for the process of energy production in cells. At this stage, we will not develop the topic of mitochondria themselves. We will limit ourselves to the information contained in mitochondrial DNA, that is, information that is necessary and sufficient for our discussion of mammalian phylogeny. Mitochondrial DNA is a molecule similar to nuclear DNA. However, it is circular and significantly shorter. It is very often used in studies of the history of genetic information changes due to its unique features regarded by the evolutionists as desirable from the perspective of identifying evolutionary developmental changes. First, unlike nuclear DNA, mitochondrial DNA does not exhibit such a wide range of difference in gene structure related to the varying functions, complexity, and traits of individual mammalian species. The nuclear DNA of different animal species may have a mosaic gene structure, meaning it can contain genes that are not present in other species. Mitochondrial DNA also lacks non-coding introns and the associated differences in their distribution. It also does not exhibit differences related to chromosomes, that is, groups of linked genes that may be passed on to subsequent generations. Instead, it possesses a relatively clear genetic code suitable for comparison, showing differences in a reliable manner through simple alignment of corresponding positions in the code. Therefore, for our purposes of analyzing similarities in the DNA code of selected mammals, we will also choose mitochondrial DNA.